For thousands of years, every civilization observed the same pattern. Children resembled their parents. Seeds from a tall plant produced more tall plants. Farmers selectively bred animals for speed, strength, or temperament. They understood the results of inheritance, but the physical mechanism driving this transfer remained completely hidden. This created a profound biological mystery. Somehow, a complex set of instructions survived the death of one generation and reliably reconstructed an entire organism in the next. The investigation into that mystery began in the mid-19th century, inside a monastery garden in what is now the Czech Republic. An obscure monk named Gregor Mendel began a methodical study of inheritance. He cultivated thousands of pea plants, but rather than relying on observation alone, Mendel counted. He meticulously recorded the traits of every offspring, generation after generation. The results revealed that hereditary traits do not blend together like paint. Instead, invisible factors seem to pass down intact as distinct individual units. As this grid demonstrates, Mendel proved that these units followed rigid mathematical rules. Inheritance wasn't random, it was a matter of probability. Mendel proved that biological inheritance behaved like information, but while he had identified the patterns, the physical carrier of that information remained entirely unknown. By the late 19th century, advances in microscopy allowed scientists to look inside cell nuclei. They observed thread-like structures called chromosomes that appeared intimately connected to reproduction. Within these chromosomes, two chemical candidates emerged as the possible carriers of hereditary information: proteins and DNA. The scientific consensus favored proteins. They were chemically diverse, and researchers assumed only such a sophisticated molecule could encode the complexity of a living organism. DNA, by contrast, appeared too simple. It contained only 4 chemical bases. Researchers assumed it merely provided structural support, playing a minor role in the shadows of proteins. This assumption dominated biology for decades, meaning any evidence to the contrary would have to be undeniable to overcome the established dogma. The first contradiction arrived when Frederick Griffith discovered a transforming principle that could transfer deadly traits between strains of bacteria. Something was moving information between cells, but he couldn't identify what it was. Years later, at the Rockefeller Institute, Oswald Avery's team systematically dismantled bacterial cells. They found that destroying proteins, lipids, and RNA had no effect, but destroying DNA stopped the transfer of traits entirely. To confirm these findings, scientists used simple viruses made of a protein shell and a DNA core to track which component entered a cell during infection. Radioactive tracking showed that the protein remained outside. The material injected inside to replicate the virus was DNA. The evidence was finally definitive. DNA was the physical carrier of life's code, and the scientific focus shifted from identifying the molecule to understanding how it worked. Knowing that DNA carried information was only the beginning. Without a structural map of the molecule, there was no way to explain the mechanism of inheritance. At King's College London, Rosalind Franklin used X-ray diffraction to capture geometric clues. Her images revealed an X-shaped pattern, indicating the molecule was a helix. Using Franklin's data and chemical constraints, James Watson and Francis Crick pieced together a model that accounted for the molecule's physical and chemical properties. Their model revealed the architecture of the double helix. It consisted of 2 strands where chemical bases paired specifically, A with T, G with C. This pairing revealed the copying mechanism itself. If the strands are pulled apart, each side serves as a perfect template to rebuild its missing half. The physical geometry of the molecule perfectly explained the ancient mystery. This structure allowed biological information to copy itself across generations. This process became known as the central dogma of molecular biology. It tracks the flow of instructions from storage in DNA through a messenger in RNA to the construction of functional proteins. Mendel's invisible hereditary units were now physical reality. Genes were recognized as specific molecular sequences within the DNA strands. Because life was now seen as a sequence of chemical instructions, biologists began applying the logic of information science to study how these sequences were stored, retrieved, and translated. Uncovering this molecular foundation provided the blueprint required to identify, analyze, and eventually modify the characteristics of life itself. By the late 20th century, the challenge shifted from identifying the molecule to reading its contents. Machines like this DNA sequencer turned the human genome into a digital archive. This ability to read the code enabled modern forensic science, targeted genetic engineering, and personalized medicine. DNA was once dismissed for its simplicity. Today, we know the human genome is a complex archive billions of letters long, written over billions of years of evolution. After thousands of years of watching traits pass through the generations, humanity has finally learned how to read its own source code.